Module 2, review of the GeneSight's tumor molecular panels. When David and I sat down and talked about this, it was really about going through all of them, and I'm going to spend the next couple of hours doing that. What I wanted to also do was give you a little bit more information to what's on the website, so that you understand a little bit about where these tumors reside, what the histology looks like. Now you know what histology pictures look like, and give you a little bit more of biology behind some of these cancers and the pathways. And as you can see from the first slide, this is the cancer pathways, every single one of them. And that's why those panels that are out there do everything, because it's basically collecting all this data and asking very simply. It's more than just sort of looking at colorectal, it's looking at lung, it's looking at prostate, breast, everything on here, all the pathways, because this is a wired network that's more complex than the car's wiring. And by the way, this is from the KEG website, so you can go on there, you can zoom in, you can look at some of the genes that are going to come up and we're talking about today. These genes, as I said, are in your module 6 at the back. Each one that's on this panel has been described, the location, the actual name in full. So EGFR1, I see that, epidermal growth factor receptor. See, I remember some of these things. But this is it. And so to dissect out each one of these pathways is really based on literature. And so sometimes there's a reason why people want to go broad to capture everything. The question then becomes, conversely, what do you do with all that information? This is a very simple story. Here's the panel, and this is what it's looking at for lung cancer. A question, by the way, Pablo, maybe towards the end, or even now if you wish, is how were the genes chosen for each panel? So I don't know if you want to talk about that now or towards the end.
Even though I'm not the best speaker about this, but the panels were selected by the reference in the literature, and every single gene that it somehow interacts and has a determinant influence in the outcome of that patient, it's related, it's documented, and that's how we build the panels. And just a bit of an example, if you go to, I think it's WebMed, and if you go into the cancer area, when you start looking for interaction of all kind of cancers, you will see that for each disease, you're going to find which are the genes involved in that disease. And our panels are more comprehensive than that, but everyone that it's inside that website, which is very well respected, are in the panels. And we added some more. Of course, you need to get to a point of a cutoff. You cannot, because if not, it's going to end up being 256 genes, and we can't do that. We can, but we don't want to. So that's the way we created the panels.
And that, by the way, will be a question I would ask you, is how did you come up with those genes, given that some of the other competitors are doing this very broad 300 gene panel. So make sure you have a good story around that that's clear and precise, exactly as Pablo had described it. All right, cancer, this is what I was telling you about. You can get mutations and then it could just be pushed away. You know, there's also stem cell cancers as well. Those are the same situation, mutation, it gets pulled away, doesn't make it. However, if you get another cell that gets a second mutation in, it could start to take hold. It could still move away, still get taken out of the system, but as these mutations start to take hold and you get a third one coming in, then you start to build up this sort of, one, the heterogeneity that you'll hear that occurs in tumors, two, the fact that they're working in concert with each other to really drive to make a nucleus for a tumor. And so by the time you get some kind of a fourth one coming in, things are really starting to take root, and that taking root starts with just the cells growing, then trying to get a blood supply in there, and so it gets richer and richer. And you know, the cells shed as well, so they could end up metastasizing, but also people are looking at detecting DNA in the blood, that as the cells break down, the DNA is shed into the system and is removed. We carry blood, broken down cells in our blood, and you're able to detect some of that. And somebody who has cancer has about 100 nanograms in 10 mils of blood of tumor DNA. And so people are able to take that and sequence it if you really are very much carrying a big chunk of tumour inside you. All right, let's start with the first one. Everyone's familiar with the skin cancer. As I said, some differences here in what I wanted to try to get to you, some pictures, some ideas about each cancer so that you have a familiarity in your head from a visual perspective what you're walking into when talking about some of these cancers. And I do recommend you read the web page. It's a very nice description of the cancers, the frequencies and all that. And I tried not to get too much into that, but something a little different that's not there that gives you an upper hand, the tools, if you like, to really have a good, knowledgeable, robust discussion with some of these folks that you're going to be, that will be your customers. So as you know, it's skin cancer. These melanocytes rise from these sort of melanomas. That's how we get brown or tanned. And clearly the sun does have an effect on the DNA in there, and it causes changes in the way the pigmentation is pulled to the top. And you can see that when you get this melanoma occurring, it goes all the way down, and there's some various stages of melanoma that really starts to describe how dangerous and deep it can go. So cautionary note here really is be careful of the sun out there, because this is what's going on when the DNA is shattered by the UV light, and the repair mechanisms within your cells are unable to repair it, and they miss something, and then that cell now becomes a dangerous cell that could turn out to create a melanoma. So I wanted to show you this, and I have this up in every single slide for every single, not every single slide, every single panel. It really gives you a sense as to where these genes reside within the genome and on the chromosomes. I think it's a kind of nice visual. Here's the panel here, and here's the location. So BRAF is one on the panel here, 7q34. I just want to give you a visual because in the appendix it just says 7q34, but what does that mean? Well, here it is. This is the P arm, this is the Q arm, and it's segmented. So there's starts off with 1 to 2 to 3 based on the staining, and there's 3, 4, and then each one of these can be broken down as well into point something. But that gives you a good sense and hopefully a visual, so that when people start talking about chromosome locations and that you are clued into it and have a visual in your head as to where these genes are located. This is basically, again, from the KEG site. Here's some of the drugs that are used against melanoma. Here's some of the pathways that come in. Again, recalling we talked about some of the genes that sit in the cell membrane that take the outside signal and go in. So you can see the NRAS mutations activate both effector pathways, RAF, MEK, ERK, and the PI3K, AKT. These are very common pathways that are used in many of the diseases in your panel here. And I know I've just thrown them out, but you can find where the BRAF plays a role. Here's the RAS-RAF pathway. Okay, and just take a little time to review these. If you can't see the picture very well because I shrunk it down, you can get access to this and blow it up. And as I say, if you go to the KEG pathway, the RAF, MEK, ERK pathway is typically activated due to the BRAF that's out of control. Okay, so it's on your panel, the BRAF, the PI3K, and the AKT pathway. Some great pictures up there that describe that. That AKT pathway is huge. I mean, it sits in the center and everything feeds into it. It's like a Grand Central Station there. So be aware of that. That's a big term you'll hear over and over again. And PTEN is another tumor suppressor gene that plays a role in here somewhere, I believe. Where is PTEN? There it is. And then, of course, the early mutations during melanoma pathogenesis, and they're typically preserved throughout the whole of the tumor progression. So you get it early, that's the one that's going to be detected. So there's going to be, it sounds like in this, and you know, melanoma is kind of an easy, not an easy one, but you can survive it. Survival rate's good. And so clearly it's one mutation that's detected, and the right treatment's given, and you're in, if you catch it early, like all cancers. Yeah?
I think these pathways, at least from what I'm seeing right now, is probably going to be a very good way for us to combat the question about Foundations Medicine doing the 300 genes, because we have the very specific pathway for melanoma, and we can say, here's our panel, we're looking at those specific genes. So, I just know for me, because that question actually came up with a potential customer of mine, is, you know, you guys are doing having you guys doing the 300 genes that Foundations is doing. So now I feel having these pathways will be better armed to answer that question. Because it just makes sense, you know, why don't we look at what is most important?
All right, gastric esophageal cancer. So this one's a bit of a nightmare. As you can see, 9th leading cancer, 5-year survival rates under 25%. That's not good at all. It is one of the deadliest, and it splits itself up into different types of carcinomas. There's a squamous cell, an adenocarcinoma, and together then you've got 90% of all these other. So you're picking up through your panel the majority of these of tying a signature, a molecular signature with a phenotype. And of course, no doubt, as always, smoking and alcohol are key players in this, especially if you are in any way susceptible to getting this type of cancer. And you can see with the picture here, the tumor are normal, how it kind of grows up through the layers and the substrata and so forth. So take a little time to review some of that. And you know, it's information that's out there in the public domain. But the more you understand about these diseases, I think it's great, and I think you'll definitely appeal to your customer that you guys know what you're doing, even from a sales force. There's nothing more, in my opinion, as a customer, there's nothing greater than a sales person who really knows their stuff, who knows the product, and can talk to you as if you're a scientist. Well, you are the scientist, but talk to you one-on-one. So really learn to understand some of these things as best you can. So some of these genes that are here, again, all these players are coming through. Here's their locations. Note, by the way, I think it's fascinating that all these genes that play in a pathway are all in different locations in the genome. And they actually, and the reason behind that, of course, is evolutionary, but they all share somewhat similar promoters, which trigger them off. So they all unfold, and that's what brings them together to get expressed at the same time. And so here's a pathway that's very complex. It's the HER. You've got the HER2 on here, which is your receptor, as well as, so I'm going to shoot backwards and forwards here a little bit, the ERCC1. And you can see that, again, these pathways with the PI3 coming up and the AKT, they're all players in the same somewhat pathway. They all worked for cell migration and cell survival. So if you change cell survival to become immortal, that's how these things take a grip. The gene expression changes, so you can see then multiple genetic alterations are key to driving this cancer. And a big set of abnormalities that come through is these allelic losses on these chromosomes, as well as the typical genes that occur in these pathways. P53 is a big player, as well as retinoblastoma and the c-MYC genes. Cyclin D1 is close down to the DNA and basically is a player in transcribing. My favorite one, just because I've worked a lot on this one, non-small cell lung cancer and small cell lung cancer. That's what these acronyms mean. So we talked a little bit about it. You smoke, and basically you have a 15% or you have an 85% likelihood of not getting lung cancer, so why not carry on? It's the 15% that are the burden on the healthcare system. And the sad part of that sort of 15%, well, not that 15%, but 90% who are the smokers, the sad part is the 10% who are non-smokers, who are women at the age of 40, that are probably predisposed. What the triggers are, no one knows, but those are the tough cases that occurs, and those are always the sad ones. But many people who smoke and get lung cancer starts 60 to 85 plus, and it's very much an old age disease, and somewhat emphasized by the smoking. So it's two types, the adenocarcinoma and squamous cell. This one here, adenocarcinoma, is found in the alveolar sacs, right at the end of the lung, which is where the exchange occurs. Basically, it's just like a big sac with blood that wraps around it, and carbon dioxide and oxygen is exchanged. there, and when those cells start to go awry, then you end up with tumours growing right on the extreme ends of the lung. And then the other one is the squamous cell, which is more centrally located, and that accounts for 25 to 30%. The small cell lung cancer, and I think this is faster growing, this is slower growing. This is sort of referred to as an oat cell cancer and small cell carcinoma. And this accounts for the rest of the cancers that are through. Oh, I'm sorry, got it the wrong way around. This one grows rapidly and spreads throughout the body as well, but this one's a slower one. But either way, both are dangerous. And since the 70s, nothing has really changed to change the 5-year survival rate. So this has a lot of attention on it. As I said, it's a real burden on the healthcare system, and it's one of those diseases which slowly, I think in the US it's like 175 are diagnosed each year, and about 175,000 each year are diagnosed, and 165,000 die. Worldwide is about 1.2 million. Here's all the crazy genes in this panel. There's a lot of them, and I've highlighted the ones, the stars. The star basically means it's the small cell lung cancer panel. This panel is under the other section, I believe, in your on your website. So, but I went through and noted that these ones are the ones that you have picked, and you can see that it's a huge number of genes because this is a big pathway, and you can see in this pathway then that there are many ways of getting to the DNA and starting to get the cell to become go into survival mode and just keep dividing. So there's the RAS-RAF pathway, you've got the KRAS gene here, you've got the FHIT, and I talk a little bit about some of these, the KRAS and the CERB. The CERB's up here. It's a receptor. And again, when you look at your package, module 6, you'll see some of these names expressed out and what they are. P53, the P69K4A, all of these are right down here. And so forth. Point mutations within the KRAS activates the GTPase activity. I hope that you looked at some of the movies. There was a real classic one that showed how when you get the mutation, when you get the EGFR1 receptor is triggered off, how the GTPase comes in and how it holds the signal, the phosphorylation, and it doesn't release it, and so it keeps sending messages to the rest of the cells that go down into the nucleus, the rest of the proteins that go down into the nucleus, and trigger off those genes that are involved in this panel and all over the chromosomes here. Okay. So overexpression then is a key player too. So if these are being overexpressed, that means there's more of them now in the cell membrane, and they're all firing off these pathways, all these pathways. So that's a big one. Inactivating mutations of P53 can lead to rapid proliferation and reduced apoptosis, which means basically cells divide, but they won't die. And then, of course, down here with the P16 Inc., I believe it's somewhere around here, inhibits formation of the cyclin D complexes and basically allows the cells to replicate. And you can see a lot of drugs are out there that are coming through. Gefitinib is probably the most famous one, as well as erlotinib, which basically are EGFR receptor blockers. And very simply, it stops the next protein coming in, which is the RAS, by sitting there on the site that's kind of folded up differently, and shuts it down. So it's a faucet. Closes the faucet. Trouble is that you get rid of all the cells that have that mutation in. If there's other cells within the tumor that have a different mutation, you get the relapse. Okay, so be aware of that. And then for the small cell lung cancer, very similar pathway, just a little bit different with some players in here. It's oncogene, the MYC oncogene. That's a big one that you'll hear a lot about. I'm trying to find it. There it is. It's further down the stream. For gene amplification, retinoblastoma gene is another player as well. And you got these loss of these tumor suppressor genes. So tumor suppressor genes are basically genes that are out there that really keep an eye on the system. And if they're knocked out, then anything goes on. It's like parents going away and saying to their kids, don't have a party in the house, right? So, and then F-HIT, as I call it, I don't think that's the right way, it's FHIT, fragile histidine triad, encodes this enzyme, and that's on chromosome 3P, which is a big cancer area, so just an FYI. All right, cancer aside, let's turn to the gist. Not gist as in the gist of it, but gastrointestinal stromal tumor. There are two types of ways in, as I will show you. There's the PDGFRA mutant one, and there's this KIT. Okay, so those are two receptors that sit there, and PDGF, platelet-derived growth factor receptor alpha, there's also a beta, but if that's mutated, then that's going to give you some of the issues in the pathway, and I'll show you that. And then the KIT, it's a C KIT, and you could see here then with the histology pictures that they're kind of spindly and long and thin, and then with the these PDGF, they become a little sort of epithelioid type, sort of roundish. And you can see with the picture here how nasty this tumor is around the stomach. It grows on the epithelial lining and it just moves into other areas, and depending on where the where it is located and close to the ascending aorta, that could prove dangerous too. There's only 3 genes in this one. Obviously, key players that the team has found to be essential here. These are receptors, and this is that topoisomerase enzyme that I was telling you about. And then I wanted to just show you the 2 different pathways, but somewhat similar, that the GIST tumor uses. If it's a PDGF receptor alpha the mutation, then it goes through the PI3 pathway and the AKT. Again, you're hearing those things come up. And the RAF-RAS pathway for proliferation and survival. And then again for proliferation and survival, it's just a slight different with the STAT3 used in that pathway if you're using the C-KIT or the KIT. And here's the basic structure of those genes and where the mutations occur in various parts. Okay, a couple of drugs out there. So you can see that the majority expresses the mutated KIT, and 5 to 7% of mutated PDGFR, no mutations. So if there's no mutations here or here, that's kind of tough. That's really it. So just interesting, 2 pathways, very similar, 2 different genes applying it. All right, thyroid cancer. Everyone's always familiar with the thyroid because of hyperthyroidism and hypo. So hyper makes you a little thinner, and hypo makes you a little fatter, depending on what's going on there as you move towards the latent periods of your life. So it's a hormonally regulated organ, or an organ that regulates some of the hormones. Sorry. You can see here it's round about the base of the throat, sits around the trachea. You can see why this is a bad one, and some of the pictures that I saw are pretty nasty. I didn't want to put any in here, but people have big bulges coming out, like a goiter of sorts, but it's pretty nasty. So it comes from these type of cells, the follicular or the parafollicular thyroid cells that are within here, and you can see how an area can get mutated, and then it can go into the trachea or even into some of these key arteries that are coming down, up and down. So not a good one to have. In this panel you have some of the key players in this pathway, including the PI3KCA. And here it is, not so complicated, it's just a very simple use of the typical cancer pathways, the RAS, BRAF, used again up here, sorry here, and then so it's really somatic rearrangements of the RET and TRK. This is another, it's just a kinase, receptor kinase, tyrosine receptor kinase, like an EGFR, which are found in particular in these parafollicular thyroid cells. The distinctive molecular features then, it really is to do with aneuploidy and high prevalence of the RAS mutation. So aneuploidy, if you recall, is chunks of the chromosome are being copied over, so you've got more than 2 copies now. So you've got 3 copies. So think of Down syndrome. If you hear aneuploidy, Down syndrome is 3 full 21 chromosomes. when you hear it, in this case, it could be just sections of chromosomes. And again, from this morning, my presentation, I was showing you how you can get inversions, deletions, copies of. That's part of what's going on here. And then the P53 gene is a big player in trying to keep this under control. So if it's knocked out, then the thyroid carcinoma can really take hold. And sodium iodide is interestingly the only drug therapy down here. Iodide, as you know, is in our salt. It's there to help our thyroids. So this is a radioactive iodine. Some of these treatments are short-lived radioactive treatments, so they don't. They have a half-life, a very short half-life, but it does make the patients very sick. Okay, rhabdomyosarcoma, one of my favorite mouth fillers. I think this one's a good one. This is really quite a rare one, found in children in particular. Soft tissue, comes from the muscle cells that have gone awry, hence the myo. And so, as you can see, there's the embryonal form and the alveolar, which is the lung, and head, neck, and urogenital tract, as well as arms and legs are some of it. And of course, these cells tend to be round, spindly, and strap-shaped, so you can see some of that going on. Again, pathologists will look at that and go, oh yeah, I recognize that one. But take a little moment to sort of recognize what some of these things look like in these histology. It's very hard to get nice, good pictures that are clearly defined. There's just a picture and it just says tumor. So I try to get some that at least showed something that would define what they meant by round or spindly. Bigger panel on this one, as you can see. Some of the same names are coming up: the KRAS, EGFR, P10, TUB-B3, c-KIT, and again all these chromosomes at work. And here's the pathway. It was a tough one to find, actually, information on this pathway and how it comes together, but it really, these genes are real players in the translocation. As you can see, this is what I wanted to show you, was that these two genes, PAX3 and the FOXO1, have come together to create a transactivating domain, a new gene. And then you've got the PAX7 and the FOXO1, again, a new type of gene. So the functionality changes. On their own, they have one function. Now it's come together. The EML4 and ALK1 was another example I showed you previously. These are players in DNA, in turning on genes at the DNA level. So you can see with these translocations, you get one that's this PAX3 FOXO1 gives you this oncogenic effect, and the other one, which is this one, really inhibiting myogenic differentiation. So stopping the cells from dying off. So this is an interesting disease, like I said, a little rarer, but there's quite a big panel on it. So it'll be interesting to see how many people ask for that one. This one's a big one, colorectal cancer, and I'm sure everybody knows somebody, and everybody who gets to the age of 50 has to be checked, but it's one of the biggest one in the Western Hemisphere. It's all about the epithelial lining, which is where it all starts. As you can see from this nice picture here, it starts right on the epithelial lining, and then it grows in and gets bigger and bigger and feeds into the blood system, and then it spreads to other organs. Notably, the liver is one of the biggest organs that gets a metastatic tumor come into it. So it's important when having your colon examined that these polyps are removed at this very early stage. So the Wnt pathway, I talked a little bit about that with the, it's funny, the Wnt pathway here is not a good thing for colorectal cancer, but for those people who had the high bone mass gene, it was a good pathway to use, because that LRP5 gene, which is in the Wnt pathway, was being slightly turned on and being used in those cell types for value, in particular bone cells. But over here in the epithelial land of the colon, it's not so good. Big panel. It's quite a big panel, as you can see. Some of the genes on this, as I said, you'll see these come up quite a bit. So I tried to get some of the other different genes that don't come up so often picked up on here. And then here's the pathway. You've got instability at the chromosomal level. You've got microsatellite instability with this one, and as well as hereditary diseases or syndromes that come in, of which we've heard about Lynch's syndrome. That's a big one. Adenomatosis polyposis. You know, Lynch's syndrome is one of those sad ones where your panel can be of great value, because it's like just having this tested and then saying, oh, look at that, you've got these genes that are involved in Lynch's syndrome. We can help you. We can, and who else in your family has that? So, because you're predisposed to it already. So I was reading an article around this one, and it's like, this is a classic value of this type of situation where the doctor doesn't understand that there's genetics at play as well as just cancer. You've just got cancer. It's more than that. So I think part of when you go to some of the secondary hospitals that don't have a strong cancer environment around them, like up in Boston or Washington, D.C. or some of the major cities, this is an important piece of information that you can drive home to them, showing your knowledge base and understanding of how genetics is really also a player to cancer and the pathways that are used, and that some of these genes that get knocked out are actually already there, and that over an accumulation of time then the presentation comes on. It doesn't mean that you're going to get colon cancer at a very early age of 2 or 3, but you will and could get it earlier at about 30, as opposed to somebody who's getting, say, colon cancer because of maybe just an old age set of circumstances. So I think that this is really important here, especially around that. Pancreatic cancer, that's a tough one. You get that, and it's not an easy one to recover from. So it's really, it's the fourth leader here. Common forms that are found in pancreatic cancer are the cystadenomas, which are non-malignant, but the other ones, the adenocarcinomas, which arise in these acinar right here, which form the part of the pancreatic duct. These are the ones that get out of control, and this is what causes the tumors. The pancreas is really not a big organ. It's about 6 inches long. It's like a big fish. It's got a big head at the front there, sits near the bile duct, and mixes in all those enzymes that are required after the food comes out of the, has been churning away in your stomach and comes out here in the very early, up at the top of the duodenum, small duodenum. So here's some pictures here, staining, that show that some of these cells are inflamed compared to a normal pancreas, and it's very much around all this, all these duct cells. A somewhat small panel, but got the usual players in here that you're starting to see and recognize coming through. And here's the pathway. Again, very similar pathways to the ones you've seen before, actually. The HER pathway, you've got the KRAS coming in. All these guys are trying to find ways to get to the DNA so that they can turn it on and make those cells survival cells. And as I mentioned, the HER2 is activated through point mutations, and then if there's another one in the KRAS, then you've got a real engaged pathway. Intermediate, so this is early stage, then some of the intermediate stages, P16 comes in, and then on a late stage you've got some of these BRCA2s coming up here, interestingly, as well as P53. BRCA1 and 2 really take part in the recombination events. They're like a big complex, and they work in that part, and that's where some of this, some of the issues occur, where they're not working to fix any of the errors. So if that's out of control or has been mutated, then it's not doing its job to fix things. Well, cancer cells love it when they have mutations, because mutations drives them, gives them an evolutionary advantage. They can start to really go crazy. So if somebody's going around fixing all those mutations, then of course that's not going to help them survive. Yeah.
So let's say in pancreatic, you do you do a sequence. Can I tell how many HER2 is being expressed, how many HER2 cell is being expressed relative to BRCA in the test?
Remember, you're looking at the genome. You're not looking at the outcome of expression of the RNA. So typically, many of these genes are upregulated, which means that the messaging from the outside is coming and say, turn it on, so the force is running big time, okay? So you're going to look at the outcome of that at the RNA level, from DNA to RNA, and people measure that. Here, you're only looking at genomic DNA. So if there has been a replication of that chunk of DNA and put back in, you can see 2 or 3 copies or 4 copies of that come up as a big tall peak in relative to somebody who has 2 copies of that mutation or that gene. So yes, so if the mutation, and this happens a lot, by the way, in many cancer cells, they do have multiple, they don't have 46 chromosomes. If you look at some of the cell lines, the tumor cell lines, they have 50, 60, 70, 80 chromosomes in there. So they have tetraploidy, 4 lots of. So so expression, be careful of the word expression using it. It's not, you're looking at genomic DNA. Genomic DNA doesn't provide expression. The outcome of using the genome to create lots of the molecules to create proteins is your expression. Unless it's been copied and dumped in again through some kind of inversion, then you can see more than 2 copies of that gene in the genome. Okay?
Yeah, I was trying to see if we could, if as a clinician, the clinician be able to say, or yeah, there's more HER2 present than there is BRCA.
The only way you can do that is to look at microarrays, which I showed you. That would be the way to turn around and say, yeah, because remember, the genome is the genome, it's fixed. How much of that product comes out is what all these players are trying to do. All these guys are trying to get to the DNA and turn it on. All right, breast cancer. This one's a big one. Everyone knows a lot about this one. The fact that it affects 1 in 8 women is pretty big and important. And so you will probably get a lot of people talking about this, some of the customers wanting to know how you guys are working with that. As you can see, it originates from the ducts, and so here's a really nice sort of pretty looking set of cells in here that have this cuboidal shape, and then when you have ductal carcinoma, it's all gone. There's no double layers, there's nothing. And this is what I was trying to find, but it was very hard to get. It's this sort of normal and abnormal, so you get to see the changes that occur at the tissue level, at the cellular level within the tissue, how this is very nice. It's got a couple of layers here, and it's all disappears, just one thin lining. So that gives you an example of what happens then in breast cancer, especially in the ductal carcinomas. This is quite a big panel. It's got some of the major players on here too, as well as BRCA. I don't know if this is a typo, but it's actually BRCA1 and not BCRA. So these are all DNA repair mechanisms, okay? This is really, so all this stuff that you're seeing here, base excision repair, down the bottom, mismatch repair, all across, this is all referring to DNA repair. What happens in breast cancer is about DNA repair, okay? The DNA is not being repaired. And so these players are key essential players in the repair mechanisms that occur in our lifestyles when cells are dividing, that sometimes it's not always done perfectly. There's backup mechanisms after backup mechanisms to ensure that the DNA is properly put together when it's copied. And so when you look at something like smoking, some of those chemicals that you inhale actually sits right in on the DNA. And in some cases it blocks the enzymes that go through and check the way it's checked. And so if there's been a mutation, it can't sometimes recognize it, and it'll just say, blow this for a laugh, I'm moving on further downstream, and it just jumps around it. Now, if the mutation's been put in there, something happened, it's jammed it up. And so these proteins are key players in checking up on that. So this is really important to get a sense of that, and these are big names that are out there that I recommend you get to recognize. P53 we talked about, P10 again, tumor suppressor genes. RAD51 is another big name that's out there, as well as cyclin D1. Okay, so again, as we go through this, you should really be hearing these names and going, oh, okay, and recognizing. But this is DNA repair, and you can see all of these drug therapies that are in there, quite a lot of them actually. Methotrexate is probably one of the most talked about ones, and its value in breast cancer, as well as tamoxifen. Head and neck, this one's a nasty one, I gotta tell you. because it really originates in those sort of moist tissue areas, and it can be anywhere in all of these areas that are highlighted on this picture of this individual here. So you can get it in all of these places. So it's one of those ones that makes you wonder how we make it through to the end of our lives without getting any of these diseases. But this one's all around the head and neck in those moist tissue areas, and you can see that these cells here are somewhat highly differentiated, and this is something that's come from the mouth. It's a smaller panel actually, given the places, but it's the same sort of cell type that's all around in the head and neck area. So these are the key ones that are players in that, and the chromosomes that they're on. And here's the complex pathway. Again, I put a couple of them up there because they were both sort of alluding to some interesting head and neck carcinomas that were coming through. You've got this RAS-RAF pathway again with the EGFR, all the way through to getting into the DNA to turn on those genes of interest, as well as over here you've got the insulin growth factor receptor, as well as EGF and other sort of pathways that are kicking on, all sending signals. Again, that's the really big story here. These signals in, again, trying to get the DNA to transcribe those genes out so that they can help the cell to survive and proliferate. Ovarian cancer, this one's interesting because really it works from the epithelial cells which surround the ovary, and so, but there are other types of cells that the cancer can interact with or start off with, initiate. You've got the germ cell carcinoma tumor up here from one of the cells, from the actual egg itself, as well as the stroma. Stroma is basically connective tissues, a stromal type of cell type, which is a supportive cell type that's in your body. And then serous is probably the most common one, 42%, and this basically is a cell type, it's epithelial, and as have many of these cancers, and they tend to, these cells let out a serum, keeps the sort of fluids around the cells so they don't dry out. And there's a whole range of other ones there that fall into different types of levels of frequency. And we'll talk a little bit about endometrial cancer in the uterine cancer. But this is quite a big panel with a lot of the genes. Again, some of the similar players that we've talked about here, BRAF, KRAS, PIK3CA and so forth. And then here is this kind of HER2, the heregulin hits off the HER2, sends in messaging into the RAS pathway, but RAS also can come over to here, onto one of these tyrosine kinase receptors, and interact with the PI3 kinase and P10, and of course our friend here, the AKT, and it's all trying to turn on these genes that help to survive and keep going. So, as you see what I've written down here, 10% of ovarian cancers are due to BRCA1 and 2, and you've heard about BRCA1. It's not about breast cancer, but it's also for ovarian cancer as well, so it can help define whether you have inherited, and that's an inheritance one, and it only represents about 10% of breast cancer, 5 to 10%. The rest are just mutations that have occurred due to whatever reason. So remember that it's not going to be the be-all and end-all of all breast cancers to utilize those genes. And so, as I just mentioned, it's an accumulation of genetic damage is the other side of the coin, which is the 90%. So some of the drug therapies that are out there as well, typical ones that tend to get used on many other cancers. The neuroblastoma and medulloblastoma cancer, it's kind of interesting, really. This is pediatrics. This is for kids. This is what kids, it really only affects kids. This one's the sad one because basically it's very aggressive. Treatment is all this, but basically there's very little that can be done. So you can have all of this stuff, but at the end of the day, it's not really going to help. This one is curable to a degree, so it tends to be, again, very invasive and rapidly growing, but it has an opportunity to be to survive from that one. So you can see the two different types of cell patterns from cells having these dark, dusty blue nuclear chromatin, which you can see in there, to them also having these rosettes, these nice, pretty looking pictures of these rosettes, and these are features typical of neuroblastoma, as well as some of this masses of small blue cells. And so many of these pathologists recognize these patterns, and it really is just patterns of cells that they're recognizing. They're not, you look at that and you go, I don't know that, but if you knew the pattern, you'd recognize it very quickly. Okay. This one's an interesting one. So there's a 1p19q co-deletion, which means in these cells, the 1p arm is gone, and the 19q arm has gone. So it's called a co-deletion. And then two different pathways that are somewhat used by these two. Again, all these pathways, all these genes, the STAT pathway is a well-defined pathway, by the way. And then over here, and by the way, it's the Wnt pathways in here too, which is over here for medulloblastoma. So different types of pathways used to give these juvenile cancers that are seen in very young children. Okay, so I mentioned to you about uterine cancer, and the uterine cancer is also called endometrial cancer, and again, it's a cancer that starts in the lining of the uterus, the epithelial cells there, and if you hear uterine, think of endometrial. I worked on this for a little bit, trying to map the gene responsible for it on the long arm of chromosome 10, 10q25. That was way back when. But again, it looks like there's more than one gene involved, typically. So cervical cancer then is sort of originates in thin flat cells on the surface of the cervix. And there's this interesting information here, the transformation zone, sort of a squamo-columnar junction where two cell types come together, and that too seems to be a very hot spot for cancers to occur in this type of in cervical cancer. Not a dramatically big panel for those two. And then this is interesting because for cervical cancer, as many of you know, is really about a virus, the human papillovirus. These are the various types, and they impact these types of genes. They integrate themselves in the E6 and E7 genes, which are very potent as oncogenes, and they dysregulate the cell cycle controls and favor the cancer cells. That's what they love. And then, of course, with the uterine cancer, you have what's type 1, which is an early age, and type 2 carcinoma, which occurs in the older age. Hyperestrogen, then that impacts endometrial hyperplasia. Some of the players in this one are P10KRAS and beta-catenin in the Wnt pathway. And then you've got over here for the older age onset of type 2 carcinoma. Here, aneuploidy, again, that copy number that I was telling you about from the genomic level. It's another piece of the chromosome that's been copied. So now you have more than one, or the cell has copied itself several times, and now it's got more than 46 or 46 chromosomes in there. P53 mutations, and of course the HER2, and here's the pathway. There's the HER2 coming in through here, as well as the beta-catenin in the Wnt pathway.
Once the mutation takes form, does it stay that way early in the cancer into metastatic? So, for example, for breast cancer, we know that there's adjuvant therapy, which sometimes might be very different from metastatic therapy. Does the gene kind of keep its character throughout into metastatic, or does it mutate even further into metastatic?
It depends what you've done to it, but typically if there's a mutation that gives it a functionality that creates survival of the cell, replication of the cell, then that's it. It's not going to change it. Now, if you get rid of that, like I said,
There are a certain kind, but then something remains.
something can remain. Now, you might hear people say, "Oh, the cancer cell is able to mutate itself." I don't know if the cell itself that has that original mutation has mutated itself, or whether, as we talk about the heterogeneity of the tumor, whether you have removed all those cells and what's left are other players. So I'm kind of favoring the latter rather than a cell who's got that mutation in, because it makes kind of sense to me that it's not, tumor cells aren't pure. Or tumor, sorry, it's not a pure set of cells, it's not homozygous. And finally, but not least, prostate cancer. So the prostate's an interesting organ, but where the cancers usually occur, and I put the 4 zones that are comprised of the prostate, it's really in zone 1 here. Most of the cancers are here, and it represents the largest volume. It's this peripheral, transition, central, and then this sort of fibromuscular, which doesn't seem to be really have any issues with cancer. You can see it sits in places where the bladder, if it becomes large, you can have bladder issues, as well as the seminal vesicle can be infected or become tumorous as well. And you can see in these nice pictures here where the cells are in relationship to the rest of the organelles around it, and how they can become tumorous as the mutated genes kick in. And there's only 3 genes here. The androgen receptor, it's on the X chromosome, XQ12, right there. For the boys only, I guess. No, actually women have the androgen receptor too. But obviously a key player, as you can see in this picture here, the androgen receptor plays a big role down here. It's normally activated by the ligand, but if its activity is in any way disrupted, then it becomes sort of, by other cofactors, then it becomes an androgen receptor independent prostate cancer, which means the androgen receptor is not doing its job anymore. It's really independent of the prostate itself. It's just gone wild. It's having a party in there. So some of the key players, the GSTP1, if that's devoid in some of these, if it's devoid, then again, that's another way that the cells can become prolific, because it's a detoxifying enzyme. And then some of the key players that we've seen, P27, P10, these regulate growth and survival. And so, of course, if you don't have these around, then the cells start to say it's time for us to have another party. So it's a very big party down there. Some of the drugs that are used, some of these ones, very unique to this particular tumor. Here's some sites that I have teased some information from that I recommend you go to. To be honest with you, NCBI, it's where everything's at. So it's very complex, it's very detailed, but it's all there, it's good stuff. And I think that takes us to the end of Module 2.